important concept in understanding monogenic systemic autoinflammatory diseases is that several of these diseases are inherited in autosomal dominant pattern. For this autosomal dominant pattern, it is characteristics that they are caused by gain of function mutations in innate immune signaling proteins. What does this mean? For inflammosomopathies, these diseases are characterized by gain of function mutations in genes encoding IL-1 activating inflammasomes. What are inflammasomes? These are large intracellular multiprotein complexes that are designed to cleave precursor pro-IL-1 beta into IL-1 beta, so highly inflammatory cytokine. And there are different types of inflammasomes. You can see here some of the recognized groups of inflammasomes, and all these are grouped under the umbrella of inflammasomopathies. The most common are pyrene inflammasome, and which causes familial Mediterranean fever, the defects in pyrene inflammasome, and the well-characterized NLRP3 inflammasome that is associated with pyrene-associated periodic fever. I will not focus on more rare inflammasomopathies. But before understanding the concept of inflammasomopathies, I would like to focus a little bit on node-like receptors, which are highly conserved cytosolic pattern recognition receptors. So these are our intracellular receptors that sense danger signals within our cells. Danger signals like some pathogens, like microbes, or damage within our cells. And if these node-like receptors sense this damage, they would start to oligomerize and activate this pro-inflammatory cascade. So this is a normal process in every human. And this is also the mechanism that is ongoing within every normal human that has no underlying mutation. The NLRP3 inflammasome and NLRP3 node-like receptor is very well characterized. On this slide, you can see that these receptors are normally in a kind of a hooked pattern, and this hooked pattern is very important that they stay in certain state of auto-inhibition or auto-repression. So they stay in a stable, dormant state. But if these receptors came into contact with some pathogens or some endogenous danger signals, they would open up, they would express their NACH domain, and they would start to oligomerize. And with this oligomerization, they would induce the apoptosis-associated speck-like protein and start the inflammasome formation with the cleavage of procaspase to active caspase, and this active caspase is a strong enzyme that contributes to cleavage of pro-IL-1 beta and pro-IL-18 in highly inflammatory IL-1 beta and IL-18. So this happens with danger signals. But what happens in persons who have underlying monogenic diseases called inflammasomopathies? So normal state is this auto-inhibited NLR protein, but with some inflammasomopathies, these patients have gain of function in oligomerization. So their NLR proteins do not stay in this auto-inhibited or auto-repressed state, but instead of this, they open up and they start spontaneous or constitutive inflammasome assembly. And you can imagine that this inflammasome assembly can start without any underlying trigger and can cause this systemic inflammation. And you will see in the following slides what are the clinical consequences of this uninduced systemic inflammation in these patients. What is important if we know the underlying pathogenic mechanisms, that most of these inflammasomopathies that induces production of IL-1 beta producing inflammasomes respond to anti-IL-1 therapy. So we have a targeted therapy for this group of diseases called inflammosomopathies, no matter what inflammasome, either this is pyrine inflammasome, NLRP1, NLRP3, or other types of inflammasomes that induce IL-1 beta production. The second group of diseases that I would like to briefly mention, you probably heard a lot of information about type 1 interference also in relation to COVID-19. These are our receptor signaling for sensing intracellular nucleic acids, either from, let's say, some viral infections or our endogenous nucleic acids, and they induces these interferon pathways, and with the production of interferons, they would bind to type 1 interferon receptor and start the YAG-STAT signaling pathway, which is involved in several cell processes of cell differentiation. And with this enhanced interferon production, they would also cause somehow constitutive transcription of pro-inflammatory cytokines. The typical diseases are Aicardi-Goutieres syndrome, which sometimes resemble a picture of systemic lupus erythematosus with different organ involvement, or STING associated vasculopathy. Or the third group of diseases are NF-κB mediated autoinflammatory diseases. NF-κB is a final common pathway for many cellular cell activating stimuli. It is involved, it migrates from cytoplasm to cell nucleus and induces transcription of several pro-inflammatory genes, and typical diseases are Blau syndrome or haploinsufficiency of A20. At the end, I would just like to briefly mention also diseases that are caused by the defects in our cytoskeleton. So different disturbances in cytoskeleton are also associated with inflammasome production and pro-inflammatory signals. In this cytoskeleton is a kind of a very branched net of different actin filaments. And I would just like to highlight one of the proteins called ARP2, ARP3, which are crucial for debranching, actually for branching of these actin networks. And with the defects of this cytoskeleton formation, we can see a huge spectrum on a crossroads between autoimmunity, autoinflammation, and allergy spectrum of clinical features. And I would just like to share one of our patients with this mutation that causes loss of ARP2/3 complex with actinopathy, with impairment in this cytoskeleton formation. And our patient presented with this huge range of clinical features ranging from infections, allergy, autoimmunity, and sky-high serum amyloid as a marker of autoinflammation. So to conclude my introductory talk, I would like to highlight that autoinflammatory diseases are a heterogeneous group of diseases. They can have several underlying mechanisms that are involved in regulation of inflammation, and they somehow illustrate the critical role of innate immunity. If we know the pathogenic mechanisms, we can also target therapies available for systemic autoinflammatory diseases based on pathogenic mechanisms. And I would particularly like to highlight the good response to IL-1 blockade in inflammosomopathies, and possibility to use YAG inhibition in type 1 interferonopathies to block the YAG-STAT signaling pathway.